against avian influenza virus and establishment of protection levels in field outbreaks in Mexico. Well, as you know, the influenza virus has a receptor on the surface. This receptor is called hemagglutinin. It's one of the receptors on the surface of the virus. The hemagglutinin has the ability to bind to acid sialic and which is present on the surface of the cell. A very important thing in this is the capacity of hemagglutinin to bind with glycoproteins containing sialic acid. And this interaction starts the endocytosis and initiate the infection process. So there is a relation between this union and the dependency of the initiate infection. Setting a strategy, and we designed a prototype product which could prevent these events. For example, we know that there is interaction, the first interaction between HA and SA, sialic acid, induce endocytosis, replication, and there's infection. But if we use a chemical interference between this and this, we could prevent the interaction. Therefore, endocytosis is not present, replication is reduced, and infection could be prevented. Material and methods. Okay, we designed our prototype of chemical receptor blocker with allantoid fluid as a source of nature protein. This protein was obtained from this virus. This is a strain that is legally used for Mexican government, H1N2. It's a low pathogenicity avian influenza virus, and it was inactivated with formalin. The complement for this preparation was a lipopolysaccharide mixture extracted for Escherichia coli, and we call it H1. We combine the allantoid fluid with the virus, inactivated virus, and the complement A1 in this proportion, 98% and 2%. And we maintained this at 40 degrees Celsius until use. Our experimental test, well, we conducted a field test in poultry farms with incidence of avian influenza virus. We know that there was a virus because we monitoring clinical things, and also we isolate and serological reaction for detection of this virus. This test serves a base for us to analyze the possibility to use this as a product. The design treatments of the field test. The test number 1 was a house beer with 85,000 beers divided in house 1, 2, 3, 4, and 5. In the case of 2, we divided in 2, 3, 4, and 5 houses. And in the same case of number 3, 1, 2, 3, 5, and 6 house beers. Okay, the application in field of receptor blocker was the following. The chemical blocker receptor was applied when we detect clinical symptoms associated with avian influenza virus. We spread application of 8 at 8 a.m. and 8 p.m. for 2 days with a 4 total application. The treatments were divided as following. The test number 1 received blocker receptor only the house 1, 2, 3, and 4. The house 5 did receive only PBS. In the case of number 2, house 2 and 3, and the house and the control was house 4 and 5. And in the same case, house 1, 2, and 3, and the house number 6 and 7 did not receive this product. Okay. We also measured the presence of immunoglobulins and IgY and IgA. In this case, we used 3 groups, A, B, and C, with 25 beers, and the control negative, our negative control was PBS. Our positive control for production of immunoglobulins was a group which received only a commercial available vaccine, and our experimental group, group C, which received the application of the chemical receptor. In the case, we measured the presence of the immunoglobulins in serum, and by 10 beers were blended in day 0, 7, 4, 40, 40, 14, 21, and 28 post-treatment. In the case of HGA, we measured the presence of the secretion, the secret immunoglobulins by virus sural neutralization. The assessment of presence of receptor blockage in chemical embryo fibroblasts was designed to test or to prove that there was a presence of this antigen on the surface of the cell. And we have two groups. The first group only received PBS. All this assay was done in chicken embryo fibroblasts. And the group 2 received the chemical receptor blocker, and we made subgroup A, B, and C. In this case, we measured the presence, or we demonstrate the presence of the antigen, the presence of the interaction between the virus and the cells by immunofluorescence and immunoperoxidase assay at 8 hours, 12, and 24 hours of application of receptor. We want to see how long does it take the interaction between the virus and the cells, and if it is possible to see this presence with this test. Our results, well, in the field test number 1, we saw that when we applied the product, when we applied this blocker receptor, there was a reduction in mortality, not in the case of number 5, which continue the mortality until the end of the project. And the case of the rest of the houses, the mortality reduces drastically. In the field test number 2, we saw the same. We saw the same effect in the field test. For example, these houses did not receive the treatment, and this house received the treatment, so there was a reduction on the mortality. In this case, also we have a reduction between these groups and these groups because we applied this product and reduced we proposed to this product as treatment, not a vaccine. Instead of vaccination, we proposed to block the virus and replication, spread the virus, stop the spread virus on the chickens. In the case of secretory immunoglobulins, we saw there was a stimulant inhibition in this case, and the group C, because we could measure by virus neutralization by tracheal washing, and we saw there was a presence of some immunity, secretory immunity. In the case of group A received PBS, there wasn't any immune response, so the group B, but we could measure some immune reaction in the group C. In the case of immunoglobulins in serum, we measured that there was, in the case of group B, which received a commercial vaccine, subcutaneously application, we measured a production of immunoglobulins as usually that we expected. But in the case of group C, we saw some immune reaction compared with the negative control. In the case of indirect immunofluorescence, we saw the following. There was a presence of the antigen. We could measure the presence of the antigen, but this reaction, immunofluorescence reaction, in the case of group subgroup A, played at 8 hours, at 12 hours, but we didn't see the presence of the virus at 24 hours. So we compared with immunoperoxidase, and we saw the same case. We couldn't see the virus or the presence of the virus on the surface of the cell at 24 hours, but we could see it at 8 hours and 12 hours. Okay, our conclusion on this job was the receptor blinding can be used can be blockaded. The receptor blocking process can be seen in influenza virus because it's been reported in other diseases like Newcastle disease. That is usually a commercial strategy. The data indicate that the duration of this event, at least, are 12 hours of effect of this product, of this prototype product, and also could be considered as a tool for control influenza virus in field breaks in other species. Okay, thank you.